Welcome students. We will be discussing on file formats for biomolecular sequences. A database is a logical coherent collection of related inherent data with some implicit meaning, and by data we mean known facts that can be recorded and that have implicit meaning. For example, the name of the gene, organism, locus, sequence, function, etc. You may have recorded these data in an indexed record book, or you may digitize and store it on a computer system using specialized software known as database management system, such as Microsoft Access, Oracle, etc. Almost all information stored in a computer must be in a file and will have file name. Each database has its own file format for storing data. A file format is a standard way that information is encoded for storage in a computer file. The file format refers to the way in which biological data are represented in the databases. Biomolecular data encompass nucleotide sequence of genes, amino acid sequence of proteins, three-dimensional coordinates of each atoms of a protein, tertiary structure, etc. The biomolecular data stored in a database can be broadly represented either as sequence or molecular coordinates. Standard file format helps us to understand and interpret the data available in the databases. Besides, different programs used for analyzing these data needs input in a standard predetermined format. Several file formats are available for bioinformatics tools and databases. Some tools have inbuilt options for converting format of a data file to another format to be made readable by other biotools so as to carry out the analysis. Based on the nature of data, we can classify file formats into two categories. 1. Sequence file formats representing DNA or protein, example include GenBank flat file, and 2. Molecular file formats representing molecular coordinates of the three-dimensional structure of a macromolecule, example include protein data bank. Some common sequence file format used in bioinformatics are GenBank, flat file, FASTA file, GCG file, MSF file format, NBRF-PIR format, EMBL format, ALN format, and PHYLIP format, etc. All entries in a database have identical format to enable computer handling of data and also to make it easier for humans. Archival databases, example GenBank and UniProt, are available as flat file that contains data in a verbose format, plain text, human readable format. GenBank. GenBank, the nucleotide sequence database format, is a standardized informational format developed by National Center for Biotechnology Information, the National Library of Medicine, and the National Institute of Health of USA, and is the standard display format for the GenBank databases. GenBank database is a part of International Sequence Database Collaboration, which is comprised of DNA Data Bank of Japan or DDBJ, in the European Molecular Biology Laboratory EMBL, and GenBank at NCBI. The GenBank format is designed to be human readable and convey both bibliographic and feature information about a sequence. The format is also parsable, that is to separate the content into more easily processed component. Each GenBank entry in the flat file format database include a concise description of the sequence, the scientific name and taxonomy of the source organism, and a table feature that identifies coding regions and other sites of biological significance, such as transcriptional unit, sites of mutations or modifications and repeats. Bibliographic references are included along with a link to the Medline unique identifier for all the published sequences. The GenBank flat file can be divided into three parts: the header, to feature, and sequence. The header of the flat file is the introductory part of the sequence format, which gives detailed information about the source organism, length and types of sequence, related literature, etc. 2. The feature part of the sequence, which shows location of the various genes present along with the related literature resource through Medline ID. And the third one, the sequence part represents sequence composition denoted by symbolically as adenine for A, T for thymine, G for guanine and C for cytosine. The word origin at the beginning of the sequence indicates the beginning of the sequence data. The end of the sequence is demarcated by a double slash symbol. A truncated version of a typical entry in GenBank database is shown here. GenBank records are delimited by a pair of forward slashes on a single line. Detailed description of each keyword identifier is given here. Locus. The locus field contains a number of different data elements, including locus name, sequence length, molecular type, GenBank division and modification date. Locus name. The locus name was originally designed to help group entries with similar sequences. The first 3 characters usually designated the organism. The rule now applied in assigning a locus name is that it must be unique. For example, for GenBank records that have 6 character accession number, example include U12345, the locus name is usually the first letter of the genus and species name followed by the accession number. For 8 character accession number, e.g. AF123456, the locus name is just the accession number. Sequence length. Number of nucleotide, base pairs or amino acid residues in the sequence record. You can submit a whole genome if you have a contiguous piece of sequence from a single molecular type. However, there is a limit of 350 kb on an individual GenBank record. The minimum length required for submission is 50 base pair or BP. Molecule type. The type of molecule that was sequenced. The various molecule type include genomic DNA, genomic RNA, precursor RNA, mRNA, cDNA, ribosomal RNA, transfer RNA, small nuclear RNA and small cytoplasmic RNA. GenBank division. The GenBank division to which a record belongs is indicated with a 3-letter abbreviation. The GenBank database is divided into 18 divisions. Example include PRI for primate sequence, ROD for rodent sequence, MAM for mammalian sequence, VRT for vertebrate sequence, INV for invertebrate sequence, PLN for plant, fungal and algal sequence, BCT for bacterial sequence, VRL for viral sequence, PHG for bacteriophage sequence, SYN for synthetic sequence, UNA for unannotated sequence, EST for EST sequence or express sequence tags, PAT for patent sequence, STS for STS or sequence tagged sites, GSS for genomic survey sequence, HTG for high throughput genomics sequence, HTC for unfinished high throughput sequence, ENV for environment survey sequence. Modification date. The date in the locus field is the date of last modification. In some case, the modification date might correspond to the release date. Definition. Brief description of sequence include information such as organism, gene name, protein name or some description of the sequence function if the sequence is known coding. If the sequence has a coding region or the CDS, description may be followed by a completeness qualifier such as complete CDS. Accession. The unique identifier for a sequence record. An accession number applies to the complete record and is usually a combination of a letter and number, such as single letter followed by 5 digits. For example, U 1, 2, 3, 4 and 5, or 2 letters followed by 6 digits. Example, AF 1, 2, 3, 4, 5 and 6. Accession number do not change even if information in the record is changed at the author's request. Sometimes, however, an original accession number might become secondary to the new accession number if the author makes a new submission that combines previous sequence. Version: a nucleotide sequence identification number that represents a single specific sequence in the GenBank database. If there is any change to the sequence data, even a single base, the version number will be increased, for example, U12345.1 to U12345.2, but the accession portion will remain stable. GI, Gen Info Identifier, sequence identification number in this case for the nucleotide sequence. If a sequence changes in any way, a new GI number will be assigned. GI sequence identifier runs parallel to the new accession dot version system of sequence identifiers. Keyword: words or phrase describing the sequence. Organism, the formal scientific name for the source organism, genus and species where appropriate, and its lineage based on the phylogenetic classification scheme used in the NCBI taxonomy database. Reference: publication by the authors of the sequence that discuss the data reported in the record. Title: title of the published work or tentative title of an unpublished work. Journal: Medline abbreviation of the journal name. PubMed: PubMed identifier references that include PubMed ID contains link from the sequence record to the corresponding PubMed record. Direct submission: contact information of the submitter such as institute, department and postal address. Features: information about genes and gene products as well as regions of biological significance reported in the sequence. These include regions of the sequence that code for proteins and RNA molecules as well as number of other features. Source: mandatory feature in each record that summarizes the length of the sequence, scientific name of the source organism and taxon ID number. Can also include other information such as map, location, strain, clone, tissue type, etc. if provided by submitter. Taxon: a stable unique identification number for the taxon of the source organism. A taxonomy ID number is assigned to each taxon, species, genus, family, etc. in the NCBI taxonomy database. CDS, or the coding sequence region of the nucleotide that corresponds with the sequence of amino acids in a protein. Location includes start and stop codons. Protein ID, a protein sequence identification number similar to version number of a nucleotide sequence and consists of 3 letters followed by 5 digits, a dot and a version number. If there is any change to the sequence data, even a single amino acid, the version number will be increased, but the accession portion will remain stable. Example: AAA 98665.1 will change to AAA 98665.2. Translation: the amino acid translation corresponding to nucleotide coding sequence. In many cases, the translations are conceptual. Note that author can indicate whether CDS is based on experimental or non-experimental evidence. Gene: a region of biological interest identified as a gene and for which a name has been assigned. Origin: the origin may be left blank, may appear as unreported or may give a local pointer to the sequence start, usually involving an experimentally determined restriction cleavage site or genetic locus if available. The sequence data begin on the line immediately below origin. Now, FASTA. The FASTA file format is the standard format for interchanging symbol sequence data. A FASTA record consists of a header line and the sequence. The header, also known as define line, begins with a greater than symbol and contains information about the sequence. The sequence is in a multi-line format, 80 characters per line. Any number of FASTA record can be placed in a single file. No special record separator is defined. The greater than symbol at the beginning of a def line indicates the start of a new record. A file with multiple FASTA record is also referred to as FASTA library. Example is shown here. Sequence representation. After the header line and comments, one or more lines may follow describing the sequence. Each line of a sequence should have fewer than 80 characters. Sequence may be protein sequence or nucleotide sequence, and they can contain gap or aligned characters. Sequences are expected to be represented in the standard IUB, IUPAC amino acid and nucleic acid codes. With these exceptions, lowercase letters are accepted and are mapped into uppercase. A single hyphen or dash can be used to represent a gap character, and in amino acid sequence, U and asterisk are acceptable letters. Numerical digits are not allowed, but are used in some databases to indicate the position in the sequence. The nucleic acid codes supported are shown here. The code supported for 24 amino acids and 3 special codes are shown here. GCG or genetic computer group format is used by the GCG suite of programs. It does look quite simple, but has two important features: the double dot, which delimits the annotation from the sequence, that is, anything after the double dot that is not numbers is understood to be sequence. The second element of importance is the check 5281. This is a checksum which is calculated for the sequence. If the sequence is edited or changed, the checksum will no longer be valid and the sequence file will not work. Here is an example of the GCG format file. Multiple sequence format or the MSF format. One can combine multiple sequence in a single file called a multiple sequence format or MSF file. MSF file include not only the sequence name, but also the sequence itself, which is usually aligned with other sequence in the file. One can specify a single sequence within a MSF file, a subset of sequence or all sequences. MSF file have the following components. File type begins with the line, is shown here below. Description, it's optional, contains informative text describing what is in the file. Dividing line, required, must include the following attributes. MSF displays the number of bases or residues in the multiple sequence alignment. Checksum displays an integer value that characterize the content of the file. Two periods act as a divider between the descriptive information and the following sequence information. Name weight must include the name of each sequence included in the alignment, as well as its length and checksum, both editable, and weight is also editable. Separating line must include 2 slashes to divide the name weight information from the sequence alignment. Multiple sequence alignment must include sequence name in the above name weight lines. This alignment allows one to view the relationship among sequences. Now, NBRF PIR format. The protein sequence data stored in Protein Information Resource database started by National Biomedical Research Foundation USA is the oldest molecular sequence database available, established in 1984. The entry arises from international collaborative efforts and are organized biologically. for example, by structurally, functional or evolutionary relationship. The entries include amino acid sequence and in many cases further annotation including citations, nucleic acid database references, current genetic information including map position and the start codon if not AUG. PIR is in a part a redundant database. Sequences are made public as soon as the database curator receives them, even before annotation or classification is verified. The database is divided into 4 sections. Section 1 annotated and classified entries, Section 2 preliminary entries, Section 3 unverified entries, Section 4 unencoded or untranslated. PIR format description. A sequence in PIR format consists of a line starting with a greater than sign followed by a 2-letter code describing the sequence type, that is P1, F1, DL, DC, RL, RC or XX, followed by semicolon followed by the sequence identification code, the database ID code. P1 stands for protein complete sequence, F1 for protein fragment, D1 for DNA, DL for DNA linear, DC for DNA circular, RL for RNA linear, RC for RNA circular, N3 for tRNA, N1 for other functional RNA and XX for unknown. Second line contains a textual description of the sequence. Third and subsequent line contains the sequence itself. The end of the sequence is marked by a asterisk character. Optionally, the above lines can be followed by one or more lines describing the sequence. Software that is supposed to read only the sequence should ignore this. A file in PIR format may comprise more than one sequence. The PIR format is also often referred to as NBRF format. The example protein sequence in NBRF format is given here. The final star or the asterisk is ignored. Biomolecular data encompass nucleotide sequence of genes, amino acid sequence of proteins, 3-dimensional coordinates of each atoms of a protein, tertiary structure, etc. The biomolecular data stored in the databases can be broadly represented either as sequence or molecular coordinates. The standard file format helps us to understand and interpret the data available in the database. The file format refers to the way in which biological data are represented in the databases. Besides, different programs used for analyzing these data needs input in a standard predetermined format. Several file formats are available for bioinformatics tools and databases. Some tools have inbuilt options for converting format of a data file to another format to be made readable by other bio tools so as to carry out the analysis. Based on the nature of data, we can classify the file format into 2 categories. The sequence file format representing DNA or protein, e.g. GenBank flat file, molecular file format representing molecular coordinates file of the 3-dimensional structure of a macromolecule, example include protein data bank format. Some common sequence file format used in bioinformatics are GenBank flat file, FASTA file, GCG file, MSF format, NBRF PIR format, EMBL format, ALN format, PHYLEP format, etc. Sequence can be protein sequence or nucleic acid sequence and can contain gaps or aligned characters. Sequences are expected to be represented in the standard IUB, IUPAC, amino acid and nucleic acid codes. Before we move on to the next session, please try to answer the following questions. Why do we need to have file formats and what are the different types of commonly used file format? Second question, what does the translational feature represent in a GenBank file? And third, define some of the features included in the GenBank file other than the nucleotide sequences. And the fourth question, what is the difference between FASTA format and MSF format? Book for your references are Algorithms on Strings, Trees and Sequence, D. Gusfield, Cambridge University Press, 1997. Introduction to Bioinformatics, Attwood and Perry Smith, Pearson's Education. Bioinformatics, a beginner's guide by Jean-Michel Claverie, John Wiley and Sons. Hope you enjoyed this session. See you next time. Until then, goodbye.